Okay, this video is about the tumor cell milieu, how to help control the tumor cell milieu. This is part one. I'll go into more later. This is an introduction to it. The main point of this video is what is the sequence of events that helps a tumor to grow, and what can you do about that? And what I'm doing is telling you the key stuff so you'll be able to understand and make sense about this when you hear other things about cancer. So here's the key point of this whole talk. It all begins with hypoxia. In my recent lecture, I talked about what is the primary cause of cancer, and it's hypoxia, which then induces the Warburg effect, whereby the cancer cell transforms itself. It becomes more primitive. It becomes like an anaerobic bacteria. So that's a transition to a primitive or vestigial, atavistic mode of metabolism, anaerobic metabolism. Okay, and so that's really a key thing. And now I'm going to show you how that affects the tumor microenvironment. The tumor microenvironment is also called the tumor milieu. And this is highly relevant because it affects cancer treatment, cancer prognosis, how likely the person is to survive. Okay, so right here is CSC. That stands for cancer stem cell. And this is a good way to picture a tumor. There's cancer stem cells in there which are more resilient, more able to replicate than the other cells, harder to treat with chemotherapy. This is one of the big problems with chemotherapy is a lot of times it'll kill all these cells in the tumor, but it won't be able to kill the stem cells. So even though the tumor will shrink down, that's called a tumor response, that does not mean the tumor's cured. These stem cells can come back. It's hard for the chemotherapy to get to them because a lot of times the center of the tumor is necrotic, with no blood vessels, and they're hiding behind that area so that they can't get the chemotherapy medication sometimes to those cancer stem cells. Sometimes they're resistant to it. They have the ability to pump it out of themselves or not interact with it. And these cancer stem cells, it's almost like a smart gang of thugs. They're able to induce the cells around them to, in a sense, protect them. And we're going to talk about how that happens. But here's the key things to know. Number one, hypoxia damages the mitochondria of the cell. It's transformed into a cancer cell, being like an anaerobic bacteria. It will then convert its glucose into pyruvate as the end of glycolysis, but the pyruvate then will go to lactate. And then the lactic acid is pumped out of the cell. So there's going to be, I'll show you a picture of what those pumps are like in a moment, but the point being the lactic acid is pumped out of the cell, causing acidosis, more protons in the surrounding extracellular matrix around the cancer cell. And then that acid, that lactic acid, has a very powerful effect. It affects everything around the cancer cell. So imagine these cancer cells in this center here are pumping lactic acid protons around themselves. That will cause suppression of all these good immune cells. So the DC here is dendritic cells. Those are a type of macrophage that will grab an antigen of the cancer and then take it to a lymph node. So it can be checked by all the other T cells, thymic lymphocytes. Here's a T cell, thymic lymphocyte cell. NK is the natural killer cells. Those are the ones that are really important for killing the cancer cells. And they can't get at the cancer because they're blocked. The cancer-associated fibroblasts, abbreviated CAF, they'll make strong fibrotic barrier in certain areas, plus the lactic acid just makes those immune cells non-functional. It's rather amazing that it's so able to do that. It's all like a synchronous response that the cancer cell basically is like a gang of thugs that just takes over the whole environment, and their ability to be hypoxic and to produce more acid is how they create all this havoc. And so what's the point? The smart move is you want to do things that optimize oxygenation of your tissues so this can never happen in the first place. And if it does happen, then don't help it. You want to avoid sodium so you don't vasoconstrict, further dropping the blood supply to this area, making it more hypoxic. Okay, this illustration comes from this article here about, targeting cancer stem cells. Hey, we're going to come back to that picture in the end when we go through a little more of the pathophysiology. Okay, so just a quick review from the last talk. Here's a regular anaerobic bacteria. Glucose goes through glycolysis, converted to pyruvate, pumps it out of the cell. A normal human cell that has oxygen available will actually prefer to send its pyruvate into the mitochondrial matrix. It runs through Krebs cycle, then it goes through electron transport along the intermitochondrial membrane, and then oxidative phosphorylation, whereby a proton is allowed back into the matrix from the intramembranous space. This is the outer mitochondrial membrane here, and it'll produce ATP. That's normal metabolism. Now here's a cancer cell, which is going to be very much like the anaerobic cell. The cancer cell here metabolizes its glucose to pyruvate, and then the pyruvate is converted to lactate, and the lactate is pumped outside of the cell. It's actually pumped with a proton, which makes this area outside the cell all acidic. And it's that acidity that then damages all the immune cells that try to get at this cancer cell. In addition, it sucks up a lot of glutamine that's also used as carbon skeletons for biosynthesis. Okay, in the previous lecture, we talked about how the fundamental transformation, the big change in gene transcription, is to make hexokinase 2 instead of the old hexokinase 1 that functions in most cells. By using hexokinase 2, it uniquely can attach to the mitochondria outer membrane, so OMM, outer mitochondrial membrane, and it will attach to VDAC, a voltage-dependent anion channel, which locks the mitochondrial membrane solid so it will not open up. Opening up in the mitochondrial membrane is what happens in apoptosis, programmed cell death. So this guy Bax right here cannot get at the outer mitochondrial membrane. Most cells will die from hypoxia, but this mitochondria will survive it. The mitochondria will no longer be able to do OXPHOS, but the cell won't die. It won't go into apoptosis. Now here is a paper, this is a good paper, Cancer Acidity, the Ultimate Frontier of Tumor Immune Escape. So what's happening is once the cancer pumps out the lactic acid, it makes it so acidic that just suppresses all the immune system. It's like superpower to make the immune system just not work. Okay, so this is really the key steps here. Anti-tumor effectors such as T cells and NK cells lose their function due to tumor acidity. They initially have a reversible anergy, but then they go into apoptosis. They die when exposed to a low pH. That means acidic environment. On the opposite hand, the immunosuppressive cells, they are able to survive the acidity. So there's more cells now protecting the cancer. Okay, so here's a picture. I have the stuff in green is what's supposed to happen. The stuff in red is the bad stuff. So let's just talk about what's supposed to happen first. You have a cancer, and then your NK cells find the cancer cells, and they kill the cancer cells. That's actually what happens all the time, every day, in everybody. Then when the cancer cells break apart, they release some of their antigens. A here is for antigen. Antigen means the recognizable component of the cancer cell. That's what the immune system recognizes to say, oh, this is a cancer cell. So typically these dendritic cells are local residential macrophages. They will then travel to the lymph node. So here would be a lymph node. Like if it was a breast cancer, this would be in the armpit, the draining regional lymph node. All right? And it'll take that antigen from the cancer cell and show it to all these other lymphocytes. That means lymph cells. And it'll try to find the ones that are able to fight that specific cancer cell and say, okay, you guys, start replicating, divide. Let's make lots of copies of yourself. We got to travel all over the human body and find any micrometastases, any residual cancer cells. This is also why exercise is so good. It gets these cells moving around real nice so they can travel all around the body, find the cancer cells, and remove them. All right, so that's what's supposed to happen. And so then you've ramped up your immune system. Clonal expansion of the antigen-specific T cells, meaning that they're specific to the antigen, meaning they recognize it, so they can then go all over the body and remove that cancer. But now here's the problem. When the cancer starts growing and pumping out lactic acid around itself, it'll suppress any immune cells that come near it. And it becomes more and more difficult to treat that cell. So that's what I'm saying. You want to optimize oxygenation of those tissues by eating a low-fat diet with no sodium added, so you maximize the amount of oxygen that gets to that cancer cell, so not as much lactic acid is going to be pumped out, and then the immune cells can better get into that area and start clearing away those cancer cells. That's what you want to happen. Okay, here's a picture of the MCT. That's a monocarboxylate transporter. It just transports lactic acid out of the cell, and that's coupled. It comes from this paper right here, lactate and neglected factor, diabetes, cancer. Okay, it also pumps out protons simultaneously, so it's a proton-coupled monocarboxylate transporter. It's called a monocarboxylate transporter because on these 3 carbon molecules, there's only one carboxylic acid. There's one carboxylic acid on lactate, okay? And same thing with on pyruvate. There's just one carboxylic acid on the 3 carbon molecule. All right, the other thing to notice is MCT4, I think of the 4 as being like an E for exporter, and that's pumping the lactate out of the cell. But there is such a thing as an MCT1. Think of 1 as being like the letter I for importer, and that can take lactate into a cell, and that's going to become relevant in just a moment. Okay, here's a paper talking about proton-coupled monocarboxylate transporters. So it really just means lactate transporters, okay, and how they crosstalk with the adjacent cells. So it's from this article in Frontiers Journal. Okay, so what's happening here is now you're going to have a symbiotic relationship. There's some tumor cells in the tumor mass that have less damaged mitochondria and have a little bit of oxidative metabolism, plus there's adjacent cells that are sort of converted to help the tumor that still have some oxidative metabolism. The point being is the glycolytic or anaerobic tumor cell will send its lactate out, and some of that lactate, because there's going to be tons of it. Remember, a cancer cell takes up 100 times as much glucose as the other cells. Some of it will get sent to these cells, and they will be able to metabolize it, okay? Because it's just a 3-carbon molecule. All of this stuff is based on glycerols. Remember, glycerol is a 3-carbon backbone of a triglyceride. It's really like a 3-carbon alkane with hydroxyl groups on it, so like a propane triol, okay, glycerol is what that is, and now you've got glycerate, you've got pyruvate, you've got lactate. They're all just 3-carbon carboxylic acids. That's why they end in ATE when they're deprotonated, okay? Glycerate, lactate, pyruvate, okay. So anyways, the point being is this lactic acid, the acid around the cancer, will transform the immune cells. The macrophage will be transformed in what's called an M2 macrophage. The little line through the O is symbolic of macrophage. And basically that makes it stink. Okay, now it can't remove the tumor. This is called immune evasion, how the cancer hides from the immune system. Okay. The excessive amount of lactic acid will also contribute to transforming the endothelial cells, the arterial lining cells in the vicinity, to promote angiogenesis. The cancer wants blood vessels growing up to it so it can jump into them and travel to other spots to metastasize. If you had cancer, you don't want angiogenesis in your tumor. You want it to just sit there and not grow and shrink away gradually, okay? CAF is cancer-associated fibroblast, and they're also doing things that contribute to the cancer. Okay, this article, look what it says about the low pH in the tumor microenvironment. It says it is a global protection shield for tumor cells. The tumor acidity acts as a broad immune escape response. It basically is like a super force field to block any immune system from getting at that cancer. It wipes out the anti-tumor cells. And it's even worse than that. It favors the tumor cells which are immunosuppressed, which actually now are protecting the cancer. So it's all screwed up. The cancer is basically like a gang of thugs that have taken over the area, and nobody can get at the cancer. And again, how does this all start? Hypoxia. That's why people say to me, why do you make such a big deal about low fat? Why do you like low sodium? Because that improves oxygenation of your tissue. Cancer does not like cancer does not like oxygenation. That's like shining the sun on a vampire, okay? That's like kryptonite for Superman, okay? That's like sunshine for Dracula. That's what you want to get that cancer out of there. Okay, this was just sort of a written summary of a lot of things we talked about. Well, we're not going to talk about iron, but those cancer cells need iron. So I would deplete myself of iron to the lowest healthy level. That's one of the things I would do if I had cancer. Being aware of this immune suppression. Remember, it all starts with hypoxia. That's what I think is the most important thing to get out of this talk. Just prevent hypoxia, and you prevent all this other stuff. Because once you go to hypoxia, there's like 20 bad things are going to happen. So just stop the hypoxia and stop all the bad things from happening. Okay, so the extracellular matrix becomes acidic because lactic acid is pumped out. Monocarboxylate transporter type 4, okay, 4 being like exporter, and that makes the extracellular matrix, ECM, acidic. This causes immune escape, where the immune system cannot now kill the cancer cells. It shuts off the NK cells and natural K cells, killer cells of the immune system. They become anergic. That means they lose their energy. That's initially reversible. Eventually, though, it'll become irreversible, and they undergo apoptosis. Cytotoxic lymphocytes, they become so-called exhausted. These are all the lingo that if you read the research papers on it, you'll now have the lingo, the jargon. Dendritic cells, we talked about those. They can't now travel to the lymph node. Their ability to do so is impaired. Your polymorphonuclear leukocytes, PMNs, they're chemotaxis. That means their ability to travel to the area of the problem is impaired. All these immune systems are messed off. And then the bad immune cells, the ones that are protecting the cancer, they're enhanced. The T regulator cells called Tregs, the myeloid, that means from the bone marrow, derived suppressor cells, MDSCs. TAMs are tumor-associated macrophages. They get transformed into M2s, meaning the bad type, which don't prevent cancer. It's like wound healing gone wrong, okay? They're protecting the wrong thing, protecting the cancer. And it's also like, where does all this come from? How can cancer do so many things? Because it's taking things that normally would be good in certain situations and making them bad. Like, you need to protect a placenta and the fetus so the mother's immune system doesn't react to the foreign cells there, okay? In a wound healing, you need to let the cells heal the wound, okay? But what the cancer has done, it's almost like a genius. It's transformed the cells around it to protect itself from actually the things that should be happening. And like, what else could you do? One of the reasons why salt is so bad is salt is sodium chloride. We talked about sodium being bad because it's a vasoconstrictor, meaning it clamps arteries down, so you get less blood to the tissue, less oxygen to the tissue. That's hypoxia, makes the situation worse. But there's even more to that. Sodium chloride comes with chloride. That's a negatively charged ion. Negatively charged ions are called anions. Well, you have to have a fixed amount of positive and negative ions in your blood. So the more negative ions you have, they will displace other negative ions from your blood. They're going to displace your bicarbonate. Bicarbonate is your buffer of blood acidity. So when you have excessive dietary chloride, you're going to displace your bicarbonate, and that's going to cause a low-grade metabolic acidosis. You see how this is all going in the wrong direction? And the same thing if you eat a lot of meat. What's unique about meat? You got a lot more sulfur-containing amino acids like methionine and cysteine. That sulfur is partially metabolized to sulfuric acid, causing a metabolic acidosis. So you see, like, the stupidest thing for a cancer patient to eat would be a heavily salted piece of meat. They're getting a double dose of metabolic acidosis. This acidosis then adds to the hypoxia of the tumor microenvironment, helping the cancer to grow. It's bad. Plus, we've talked about how the unique amino acid composition of the meat, having more leucine, lysine, and methionine, that is basically like miracle grow for cancer, like a growth hormone on the cancer. Plus all the fat inducing insulin resistance cranks up insulin. Insulin's also a mitogen. Mitogenic means that it promotes cell division. So you want to stay away from that. You want to be plants only. The other thing too, look at your fruits and vegetables, okay? They're really alkaline. That's good. Alkaline is your friend. It goes against the cancer. You want to control your psychological stress. You don't want any unnecessary psychological stress. Some excitement of exercising, positive social interaction, that's all good. Attention, that's all good. But excessive psychological stress, caffeine, etc., that's all bad. So excess stress contributes to increasing MMP, matrix metalloproteinase, which cuts a path through the extracellular matrix to facilitate the cancer escaping out there. So it's a little bit confusing, and it almost sounds partly contradictory. You can get a fibrotic response built around some of your stem cells that protects those stem cells and makes it harder to actually eradicate them, but you can also get areas of the tumor environment where there's a cut open path by these MMPs, matrix metalloproteinases, that facilitate cancer spread to other locations. And then they get covered in the platelets, which are activated by psychological stress, caffeine equivalents, and that kind of also hides them as they travel from the immune system. But remember, it all starts with hypoxia. Hypoxia and acidosis are the friends of the cancer. They're not your friend. Okay, so, and then if the cancer cells are protected, the cancer can recur even if most of the tumor shrunk down. That's why sometimes when people talk about chemo, they'll say the patient had a good response. But a good response does not mean cured. A good response means the tumor shrank a lot. But what you want is a cure, which means that the cancer stem cells have to be gone. And that's not as easy to accomplish. It tends to work pretty well in testicular cancer, but not as much in a lot of these other solid tumors. Okay. Also, hypoxia downregulates expression of an antigen on cancer cells called the MHC, major histocompatibility complex antigen type 1, and that makes it so the T cells can't recognize the cancer. So all these things are going in the wrong direction with hypoxia. Okay. And you don't even need any mutations. This is not somatic mutation theory. This is the metabolic theory of cancer, and guess what? It makes a ton of sense and it connects everything. Okay. You need to know metabolic theory of cancer if you want to, understand this stuff. Okay, anaerobic metabolism causes relative shutdown of forward Krebs. We talked about that before. Oh, when you shut down the forward Krebs cycle, which we talked about as an electron harvester for electron transport and oxphos, what also happens is you're no longer going to be, decarboxylating. When you run forward, it's an oxidative process, Krebs cycle, and it's separating out CO2s. When you stop running Krebs cycle forward because of anaerobic metabolism in the cancer cells, you're making less CO2, and that drop in CO2 means less vasodilation, more because the arteries are constricted down because of this lack of CO2 to vasodilate them, more vasoconstriction, more hypoxia. So that's how the cancer cell, it gets the whole game going in its favor. And what I would do if I had cancer is I would want to go low fat, low vegan, no salt added, and a lot of exercise, get more oxygen going to my tissues, get those lymph cells, those WBCs moving around. Okay, we talked a little bit. I gave a lecture recently about the effect of stress on cancer. I'll just show one of those slides again. So this is just the point that all the good guys are going to be suppressed by the lactic acidosis. The natural killer cells, the dendritic cells, the cytotoxic T cells, okay? And then the bad ones, the T regulator cells, tumor-associated macrophages, the M2s, the myeloid-derived cells, those are all being suppressed. And so here it is again. Almost it's like the king of the thugs are the cancer stem cells, and they're hiding out, and it's almost like this, these are like sandbag barriers of fibrotic cells protecting them, so it's hard for anything to get to them. And what I would do is you think about if you run water over a rock long enough, it'll start to erode the rock. So you just want good oxygenation coming in every day, lots of exercise so the immune cells can move all over the place. That exercise, when the muscles contract, it contracts the lymphatic vessels. So it keeps the WBCs, the white blood cells, circulating, and they can then chip away at that cancer. They can then travel around the body and remove it. That's what you want. Optimal good blood flow, so you can eradicate these cancer stem cells. And you can at least keep the darn thing suppressed, and it's not getting any extra vessels, because the hypoxia induces the ingrowth of new vessels. That's called angiogenesis, tumoral angiogenesis. And that enables the cancer to spread to other areas. So you want to keep those cancer cells trapped in one spot. Say, okay, I maybe can't get into your bunker, but you ain't getting out. All right? And that's what I would do if I had cancer to kind of control this process. Optimize oxygenation of the tissues. Avoid anything that's going to cause decreased oxygen, meaning, sodium vasoconstriction. Avoid things that are going to cause acidosis, like sodium chloride, animal protein, etc. So I think that's the last slide here. Yeah, that's the end. So anyways, hope that was helpful.